Hi, thank you for the invitation. I'm going to be speaking to you today about some of my research work in autism with an emphasis on bridging the gap between genetics and patient-centered research. My autism research has been focused on three areas: subtyping, females with autism, and gene regulatory factors. First, I will briefly talk about few examples of genetic research studies, and then I will discuss how I'm trying to use lessons that I have learned from basic research to develop an integrated concept called autism genetics outcomes or OUTGO. In addition to mutations in DNA coding sequences, abnormalities in gene regulatory processes, including alternative splicing and non-coding RNAs, may also contribute to complex conditions such as autism. Coding sequences of genes are composed of several segments called exons. Alternative splicing is a process by which different assembly of exons may be made, resulting in generating different versions of a gene. Non-coding RNAs, such as microRNAs, do not code for proteins, but may bind to the protein coding genes and regulate their expression level. During the last several years, significant progress has been made in identifying genetic causes of autism, with the exception of a few rare mutations identified as causative genes. Majority of the findings are common genetic risk factors with unknown function. A GBOS study more than a decade ago identified a genetic variant or a SNP on chromosome 5p to be associated with autism. This was an important discovery, but the associated SNP was outside the coding sequences, and its functional role in autism was not clear at that time. A few years later, after that GBOS discovery, a research team led by Dan Campbell used a novel approach by utilizing bioinformatics tools, sequence homology, and gene expression, and showed that this 5p14 autism-associated SNP is located inside the non-coding RNA, which has sequence homology to an X-linked protein coding gene. The non-protein product produced from this long non-coding RNA binds to the protein coding gene on X chromosome, resulting in downregulation of this X-linked gene. This discovery demonstrated how genes located on two different chromosomes may interact with each other and potentially contribute to the etiology of autism. Different approaches have been used to address phenotypic heterogeneity in autism and to identify subtypes. Many of these studies stratify subjects based on severity along a single domain, such as language impairment, while individuals with ASD manifest deficits across a broad range of behaviors. ADIR is an assessment tool considered as a gold standard for assessing autism spectrum disorder symptoms. It consists of approximately about more than 100 questions. Valerie Hugh evaluated ADIR scores across a broad range of symptoms using multiple clustering methods. This holistic approach resulted in identification of 4 subgroups of autistic individuals, shown here by 4 different colors. In collaboration with her group, we used the identified ADIR subtyping to reanalyze genotyping data available from about 400 multiplex families. Multiplex means they have more than one affected child. To do so, we developed a multiple-step stratification method and generated 16 ADIR-related subsets, as shown in this figure. and ran separately linkage analysis for each one of these subsets. Obviously, subtyping reduced sample sizes, and statistically speaking, we would expect to lose detection power in analysis. However, since we actually lowered the existing heterogeneity in the study cohort and generated more homogeneous subtypes, our linkage results improved. We find subtype-specific linkage and two subtypes at the bottom of this stratification pipeline, with sample sizes as small as 8 and 15 families showed positive linkage to chromosome 5p14. It should be noted that linkage for the original non-stratified group of 392 families did not result in any significant LOD score in this analysis. These two subtypes with a positive linkage to 5p14 may further refine the reported association between the long non-coding RNA in this region and autism. Now I'm going to talk about another project. Females with autism have two X chromosomes. X chromosome inactivation occurs early in embryonic development in females to achieve gene dosage compensation with males. This is a random process, and usually one of the two X chromosomes in females is randomly inactivated. Deviation from random X inactivation, X chromosome inactivation, or skewness sometimes may happen, and it may have impact on expression of X chromosome genes in the females. Autism is more common in boys than girls. To assess if X chromosome inactivation contributes to the observed sex ratio differences, we assess X chromosome inactivation pattern in autistic females compared with controls. We found a higher level of unequal X inactivation or skewness in autistic females versus controls, 29 versus 11% in controls. X inactivation is a DNA methylation process. In the next slide, I will show how we use the X in chromosome inactivation status as a stratification factor in evaluating expression of a gene involved in circadian rhythm. Circadian rhythms are biological clocks regulating physical, mental, and behavioral changes in response to a daily cycle. This intrinsic system influences body functions such as sleep-awake cycles, eating habits, and digestion. Abnormalities in these biological processes have been reported in autism. Key genetic components of circadian rhythms are chromatin remodeling genes that regulate DNA methylation. Using a combination of alternative splicing and sequence homology, we identified a variant for KDM5C, an X-linked chromatin remodeling gene with a novel 3' UTR end that is originated from a long non-coding RNA. We showed that this long non-coding RNA is part of a transposed gene with a sequence homology to a parent gene on chromosome 10. Using Tagman assays, we showed that expression level of this novel variant is significantly higher in autistic females with an extreme pattern of X chromosome inactivation, or so-called XIS. This finding is important because chromatin genes are involved in DNA methylation, and X chromosome inactivation, which we use as a subtyping method in this study, is also DNA methylation dependent process. In my opinion, this study further demonstrates that the importance of using biologically meaningful subtyping in autism genetic studies. To summarize the first part of my talk, I would like to highlight 3 points. Number one, ASD has a complex etiology that includes both genetic and environmental risk factors. And because of very diverse clinical presentations, subtyping may be needed to improve study results. Obviously, these are real challenges in studying autism, but they also provide opportunities to create novel approaches and establish multidisciplinary collaborations. In the second part of my presentation, I would like to discuss the importance of multidisciplinary approaches with an emphasis on making a bridge between genetics and patient-centered research. I think making a practical connection between these two types of research may facilitate improving translational aspects in autism genetics. Despite significant progress made in understanding the etiology of autism, there are no effective biomarkers to facilitate the early detection and effective treatment. Two strategies have been commonly used to address these barriers: increasing sample size in research studies to improve statistical power, and developing new analytical tools. These strategies have been helpful in some cases, but translation of research from bench to bedside is still a major challenge. I would like to share a story that triggered my interest in finding a way to incorporate community participatory research or patient-centered research in autism genetics research. In 2009, I developed a database to archive autism susceptibility genetic risk factors, including candidate genes, CMVs, fragile sites, and non-coding RNAs. One day, I received a call from a father of a son with autism. He asked how he could use my database to analyze his son's genetic data that he obtained from a direct-to-consumer genetic company to understand the causes of his son's condition. I explained to him that, unfortunately, it is not possible to use any database to solve such a complex question. This unexpected conversation had a profound impact on me and encouraged me to start thinking about how to promote building partnership with patient advocates to make research more patient-centered. Patient-centered research is an emerging new approach to research that is focused on the needs and perspectives of patients and their families. This approach has been promoted by funding agencies, including PCORI. Although I am trained as a genetic researcher, after learning about patient-centered approaches, I was inspired to see if I could apply my expertise to make a connection between these two disciplines. To address this need, I developed an engagement model called OutGo, with an emphasis to facilitate incorporating patient perspectives when developing a genetic research hypothesis. First, we formed a community advisory board. We used a variety of communication methods for feedback collections. We shared information about different topics, including outcomes research, genetics, precision medicine, electronic health records, and ethical issues, and reviewed a few examples of patient stories, relevant publications, and resources. We collected and processed feedback to identify the barriers, facilitators, and needs as shown in this figure. We also identified a list of recommendations, including the need in raising awareness in the research community, the need to develop disease-specific examples. And we choose autism to develop condition-specific examples and for the refined engagement and implementation protocols that we have developed. We identified that one key barrier is the lack of interactions or communications between researchers from these two disciplines. So first, we focus on so-called conceptualization to create a hybrid concept connecting the outcomes and genetics and disciplines, and to demonstrate the potential and benefits of interplay between these two disciplines. Our goals were to stimulate interest among different stakeholders and identify practical ways to incorporate patient or parents' perspectives when designing genetics outcomes or geo studies. We refer to integrated research approaches that utilize data and principles of both genetics and outcome research as geo-hypotheses. Traditionally, genetics research studies rely on quantitative methods. On the other hand, qualitative methods are most commonly used in outcomes research. In genetic research, we typically test a hypothesis using scientific data to find disease etiology. In outcomes research, hypothesis generation is often the endpoint, and it is finalized by incorporating patient perspectives. Despite differences in their approaches to hypothesis generation, data collection, and overall objectives, both genetics and outcomes research aim to make research more translational by improving health outcomes or contributing to precision medicine. In the OutGo initiative, we attempted to show that there is a need to incorporate patient or parent perspectives in study design, and that using mixed methods may facilitate, at least in theory, moving the autism genetics field closer to the center of the, meaning closer to the translational research. But why do we need to incorporate patient perspectives? Let me briefly describe two examples to address this question. Example one is that parents have frequently raised concern about GI issues in autism. A mother of a boy with autism first brought attention to a potential connection between her child's rapid developmental and behavioral regression and his GI symptoms. She had no formal medical or scientific background, but started to study medical literature in a desperate attempt to find a solution that would ease her child's condition. In 1998, she published a perspective paper. This and similar concerns raised by other parents eventually led to increased research on the co-occurrence of GI issues in autism. In 2009, Campbell et al. Identified the association between a variant in a gene called MET in families with co-occurring autism and GI conditions. This gene has a dual function. It contributes to both GI functions and brain development. Subsequently, more attention after this discovery has been given to the importance of treating GI problems in individuals with autism, particularly for non-verbal cases. Second example is about fever effect. For many years, the parents of children with autism have reported that behavioral symptoms diminished when the child had a fever. A study published in 2007 reported that some children with ASD may show behavioral improvements during fever. Subsequently, functional and mice studies resulted in discovering the cellular mechanisms of this so-called fever effect. Later, the results of a small clinical trial showed that a nutritional compound may improve symptoms of autism, at least in some cases, by mimicking what happened in the body during a fever. So I think by looking at these two examples, I could see two common elements. In both examples, the discoveries resulted in improving patient health outcomes. And the second one is that the awareness about GI issues and fever effect initially noted by parents. Obviously, not every anecdotal observation has a biological or mechanistic basis, but some do. And how could we develop frameworks to facilitate such connections so they don't happen just by chance, or we don't have to wait several years to make the connection with research discoveries? To address this need, we developed a semi-structured literature review process to collect and process feedback from our study participants to evaluate research findings in the literature from different perspectives and identify potential geo-hypotheses that may arise from these findings. The process included defining inclusion criteria for the literature review, applying these criteria to select papers for team evaluation, and implementing a two-step evaluation process, and tiers 1 and 2, to obtain members' feedback. To further streamline the process, a uniform template was developed and used for summarizing each topic to facilitate the contribution of all members, regardless of their background and expertise, as well as an evaluation metrics and scoring system. This workflow was exercised by OUTGO team members to formulate a geo-hypothesis for one topic. Using the inclusion criteria, 7 ABA advisory board members recommended 60 papers, from which 10 potential geo-hypotheses were identified for the topics listed in this figure. Participants elevated 3 of these topics to for further consideration. Subsequently, depression in autism was selected after tier 2 evaluation. And finally, the expert panel drafted a geo-hypothesis for the selected topic, which was then further refined and finalized by contribution of community stakeholders or OUTGO team members. Lessons learned from this exercise or this engagement protocol included: we learned that there is a great need in recognizing the importance of developing a conceptual framework and also assessing literature from the geo-concept perspective. The third lesson was recognizing the importance of educational geo-examples. And the fourth one, developing and employing synergistic strategies for patient-centered initiatives. We also identify some challenges and opportunities. Traditionally, outcomes research focuses on identifying and prioritizing patient needs. However, addressing those needs may be beyond the scope of genetics research. So this is a major challenge. This incompatibility has been one of the key obstacles that prevented the formation of such partnerships. One solution we thought to address this challenge is to build partnerships based on flexible and mixed methods, such as actionable geo-hypotheses. The developed semi-structured literature review addresses this challenge by retrospectively synthesizing research findings from the literature, identifying potential future direction with a focus on geo aspect that might arise from these findings, meaning that they are more likely to be actionable, and prioritizing them with input from patients and other stakeholders. We used a series of community engagement efforts, both for educational and dissemination purposes. These efforts resulted in identifying additional resources and drafting a strategic planning for sustainability, which resulted in securing 2 capacity building PCORI funded projects. Project 1, or as we call it, MARTH, aims to develop a research agenda and strategies for a regional consortium to promote patient-centered outcomes research, such as GEO projects. And Project 2, or Data Science Project, aims to develop a roadmap for promoting such studies using secondary data analysis of existing databases. While these 2 projects are separate, we strive to find synergy whenever possible. For example, we have developed a feedback loop process whereby the data science team provided inputs on the feasibility of using secondary data analysis to address research questions identified by the MARC team. Autism is a complex and heterogeneous condition requiring innovative and integrative approaches to research. We need to evaluate big data through different lenses and perspectives, which I call the human factor. While state-of-the-art technologies are essentials, we should also emphasize access to state-of-art conceptual frameworks and educational curricula to promote patient-centered autism research. Newborn screening system is an excellent ecosystem to further define roadmaps and protocols for narrowing the gap between genetics and outcomes research because of its multi-components and multi-stakeholder nature. I think the work that I presented has the potential to contribute potentially towards the progress of rare diseases research by advancing our understanding of neurodevelopmental behaviors in rare disorders, including RASP conditions, by facilitating linking patient perspectives, either stories or patient-reported outcomes to research discoveries, and by streamlining implementation of patient-reported outcomes in research hypothesis development to accelerate the development of new treatments and interventions. I would like to thank the families and patients and collaborators from multiple institutions that made these collaborative efforts possible. And my research works have been funded by Autism Speaks, Department of Defense, BioNexus, Casey, and PCORI. Thank you.